Today in this session, let us discuss about gluconeogenesis in a very brief manner. So gluconeogenesis, as you know, is a process or pathway in which glucose are synthesized from non-carbohydrate compounds like lactate, glycerol, pyruvate, and amino acids. So if you look at this flow chart, you can see that this is a flow chart of glycolysis. So here you can see glucose, yeah, and glucose is ultimately converted into pyruvate, the end product pyruvate. Okay. So if you see the steps of glycolysis, there are 10 steps, and out of these 10 steps, almost all 7 of the steps are reversible and 3 steps are irreversible. There are 3 irreversible steps. The first step catalyzed by hexokinase or glucokinase is irreversible, and the third step catalyzed by uh, this one, the phosphofructokinase, is irreversible step, and the third irreversible step we have is conversion of phosphoryl pyruvate to pyruvate by pyruvate kinase enzyme. So in gluconeogenesis, it is the synthesis of glucose from non-carbohydrate sources. So if you see, and as we have seen the substrates for gluconeogenesis, what are the substrates? Substrates are uh, like your lactate, pyruvate, then the glycerol, then amino acids. Amino acids, or you can also say propionyl. Propionyl CoA. These are the different non-carbohydrate substrates from which glucose can be synthesized through the process of gluconeogenesis. So, gluconeogenesis is not just a simple reversal of glycolysis. So, if you see how to overcome this pyruvate cannot be converted to phosphoryl pyruvate directly. But how this pyruvate can be ultimately converted to phosphoryl pyruvate, let us see the steps in detail in the next slide. Here we are going to see how this pyruvate is ultimately converted into phosphoryl pyruvate. Okay. So pyruvate, the process of glycolysis takes place in the cytosol, so pyruvate is formed in the cytosol. And this pyruvate through this transporter, that is monocarboxylate transporter, that is a specific transporter for pyruvate, this pyruvate can enter into the matrix of the inner mitochondrial matrix. So pyruvate is now in the inner mitochondrial matrix. And in the next reaction, pyruvate is converted to oxaloacetate by enzyme pyruvate carboxylase. This is a biotin-dependent enzyme, and carbon dioxide molecule is needed for this reaction, and a molecule of ATP is required. ATP is converted to ADP. So you get oxaloacetate. This oxaloacetate is again, it cannot come out of the mitochondrial it cannot pass through the mitochondrial inner mitochondrial membrane. So uh, it needs to be converted into malate. Next compound is malate, and malate, this reaction is catalyzed by malate dehydrogenase enzyme, and in the process oxaloacetate is reduced to malate. And this malate has got its transporter, that is called dicarboxylate transporter. Through this transporter, malate can come out into the cytosol, and in the cytosol, the reversal of the reaction, reversal of this particular reaction takes place, and malate is converted into oxaloacetate. Okay. Now this oxaloacetate is again converted into phosphoryl pyruvate by enzyme phosphoenol pyruvate carboxyl kinase, GTP molecule is utilized and a molecule of carbon dioxide is released in this reaction, and you get phosphoenol pyruvate. Now, other reactions after this reaction phosphoenol pyruvate, rest of the some of the reactions are reversible. So next irreversible step is conversion of fructose 1,6 bisphosphate, fructose 1,6 bisphosphate to fructose 6-phosphate, fructose 6-phosphate, and this is done by 1,6 fructose 1,6 bisphosphate, fructose 1,6 bisphosphatase enzyme. And then the third irreversible step is conversion of glucose 6-phosphate to 3-glucose, and this is done by glucose 6-phosphatase enzyme. So ultimately the glucose is formed. So these are the three irreversible steps of glycolysis. And if you see on the left side of this slide, I have given here how this different amino acids can be converted into intermediate TCA cycle, like alpha-ketoglutarate, and how this valine isolation vitamin is converted into succinyl CoA. Intermediate TCA cycle again, phenylalanine tyrosine can be converted to fumarate, aspartate can be converted to oxaloacetate. This way, this several amino acids, both ketogenic and glucogenic amino acids. The ketogenic, when I say those amino acids which can be converted to acetyl CoA are called ketogenic amino acids, or those amino acids which can be converted to acetoacetate are called ketogenic acetoacetate. So these amino acids are called ketogenic amino acids. So both ketogenic and glucogenic amino acids can be ultimately converted into glucose by the process of gluconeogenesis. So these are the important questions: irreversible steps of glycolysis, and this how these different amino acids can be converted to intermediate TCA cycle, and how they can join the gluconeogenesis pathway. And you have to remember those non-carbohydrate substances that can contribute to the formation of glucose. And there was also, I forgot to mention, glycerol. Glycerol can also form carbohydrate, but for if it is glucose from glycerol, you need to overcome only two irreversible steps. The first two irreversible steps, the steps catalyzed by phosphoenol pyruvate, phosphoenol pyruvate to pyruvate, that is not required. Okay, it is bypassed. Thank you.